So with interferons, we're talking just about viruses. Okay, this picture, if it doesn't make sense already, this is a host cell, right, in your body, we could say your finger, and these are the cells surrounding it. Okay, I'm just going to draw parts of them. Okay, so a virus enters a host cell, right? And when he enters the host cell, what does he do? He encodes, right?
Yes.
He lets out his genetic material. And that genetic material will make babies that will then go infect the other cells around it, right? Now imagine if you're this host cell. It's a nice, beautiful day, just loving life. And then all of a sudden you look out the window and you see long double-stranded RNA floating by.
Mm-hmm.
Is that normal? No. What does that mean?
You're infected.
With a
Virus.
virus.
If this is inside the cell, then
Yeah, it'll be in the cytoplasm.
Okay.
So if you are a host cell and you see long double-stranded RNA, that is bad. That means that there's a virus.
It's double-stranded because usually it's just one-stranded?
It's mainly the long.
The long.
Yeah,
okay.
the fact that it's really long.
Okay.
Wait, so why are you putting double-stranded RNA?
Because that's just what triggers it. Because we have RNA in our bodies, right? But it's usually in the single-strand form and it's usually short.
Okay.
So the fact that there's long double-stranded RNA, the reason why I'm saying this is the trigger for interferons, long double-stranded RNA.
But a lot of our viruses are all single-stranded.
Yeah, we're going to talk about it. Don't worry about it. Okay. So the nucleus sees long double-stranded RNA, and what is he thinking? I'm toast. Okay? This virus has infected me. I'm going to die. Okay? So he knows he probably has like an hour left. Instead of being selfish and like crying about it, he decides that he's going to make something of it. He's going to go out strong, right? So he knows when this virus kills him, where is that virus going to go?
Next host.
To the neighboring cells, right? His friends. And he likes his friends, and he doesn't want them to have the same fate, okay? So what he does is he sends out, it's another cytokine, and they're called interferons. Interferons are small antiviral proteins. Now, what do you think he's telling the cells around them to do?
To like get like spark of their defenses.
Yeah, so why did the virus enter this host cell in the first place?
Replicate.
To replicate, because it needs the host cell's cellular machinery, the ribosomes, in order to replicate. If the virus breaks out of these cells and goes to the others, it wants to use the ribosomes that do protein synthesis in the other cells. So interferons go to the other cells and they tell them, shut down protein synthesis. Okay? Because if they shut down protein synthesis, the virus, they exit, they go to the cells, and what do? If there's no protein synthesis happening, they can't do anything.
I'll just keep going.
They'll just move on. Or they'll get stuck in there and they'll open up, let the nucleic acid out, and it can't do anything.
Do you know how big of an area like that?
It's
spread?
localized. Because imagine if it was body-wide, all of your cells just shut down at once. That'd be bad, okay? And then also this is temporary. It's not a long-term thing. Because if those cells stop making proteins altogether, eventually they die. So it's short and it's localized. Now, would this happen for bacteria?
So it's just viruses.
Only the viruses will have this long double-stranded RNA. And then also, if you shut down protein synthesis for a bacteria, does the bacteria care?
Probably not.
No. It doesn't need the host cell's
Yeah,
protein
it's producing
synthesis.
its own.
It does binary fission on its own, right?
Yeah.
So now to your question. A lot of people in the final last year got really caught up. You know how we have positive RNA, negative RNA, double-stranded DNA, and retrovirus? They thought only RNA viruses could stimulate interferon response. All viruses can. Even a double-stranded DNA virus, when it comes in, it's going to make a lot of different forms of nucleic acid when it comes to making its proteins. And long double-stranded RNA is almost always an intermediate. Okay? So like double-stranded DNA, remember, it'll go in the nucleus, and then it will make RNA through transcription. And then in order to replicate it, it will make double-stranded RNA in order to replicate it. So no matter what virus it is, it's always at one point during its infection inside a host cell going to make long double-stranded RNA. So it doesn't matter if it's a retro, positive RNA, negative RNA, double-stranded DNA, they can all trigger interferons.
It's like after the proteins are made and they see that long double-stranded
It depends on the virus when they would see it. But at one point, it's going to be, long double-stranded RNA is an intermediate for almost every virus.
Do you need to know that?
No, the only reason I bring it up is because last year on the final, people were answering that only RNA viruses could trigger interferons. And I'm just trying to, it's not going to be like a question, I just don't want you on the test to think, oh, only the RNA viruses could do it. Because he's not trying to trick you if he's talking about a double-stranded DNA virus and interferons. As long as it's a virus, it's going to eventually make long double-stranded RNA while it's in this cell, and interferons would be produced.
When is the long double-stranded RNA made?
It depends for each virus. So positive RNA viruses, they'll make it it's usually when they're replicating their RNA. So they have single strand, and in order to replicate, they make it double-stranded real quick, and then they separate it to two. Does that make sense?
It's just for quicker replication.
Yeah, it's because in order to make new DNA, you have to have a template strand. Remember we talked about PCR? You have to have something to build off of.
Okay.
So it doesn't happen with bacteria. The biggest thing, they shut down protein synthesis in the neighboring cells. Interferons are antiviral proteins that shut them down.
Does this trigger anything else? Like
Not that I know of.
T cells or
Okay,
anything
so like if they go in there and they see like, oh, they can't use the protein synthesis, do they just leave? Or like, because our cells will eventually die if they're just
Well,
sitting there and not.
think about it like we were saying before, it will be quick. It's not a long thing. But when this virus comes in, it's not like he gets in and says, ooh, is protein synthesis going on, and then uncoats. He uncoats when he gets in there. Once his DNA's out and he finds out that there's no protein synthesis, we're going to have, let's say that this cell right here isn't carrying out protein synthesis, because let's say this cell sent out the interferons. We have enzymes. If that DNA or RNA is floating in there too long, we'll chop it up. So by the time that the protein synthesis turns back on, it's like nothing ever happened. Does that make sense?
So then it's cytal to the bacteria.
Cyto to the
To
virus,
virus.
but we don't really like to say cyto because they weren't alive in the first place. But what you're thinking is good. Pretty cool, huh?
That is
I think
cool.
it is cool.
Really cool.
Pretty smart.